Hey guys, what's going on? So today we are going to be talking about my work, right? So a little bit ironic that I haven't really been posting much on my work yet. It's not because I don't like it, it's because I have been thinking about how I want to present it. And so with that being said, I am gonna just kind of jump right into it, right? I have been thinking about how I want to organize things, but I think the best way to start is to just jump in head first. Before I do that, disclaimer: if you like this kind of content, you can hit that subscribe button, hit that like button, and you can visit my Patreon page. Anyways, let's get on to the topic, right? So the topic is ionotropic glutamate receptors. I guess the first thing we would like to do is kind of just decompose this name, right? Because the name is everything, right? So what does ionotropic mean, for those of you who don't know? What's glutamate, right? And what's a receptor, right? So these are good questions, right? If you have questions, these are good ones, right? So maybe in this first video, we will cover these few questions here, right? So ionotropic, what does ionotropic mean? Well, there's two, we can contrast that with another term, right? So ionotropic versus metabotropic. So when we contrast these two terms, right? So metabotropic means that this term refers to the idea that you have some cell, right? So here's our cell. And actually, I'm gonna erase this really quick. You're gonna have a cell, do it in green. And it's got a bilayer, a lipid bilayer. Let's see if I can do a decent enough circle. So this is our lipid bilayer, and say inside of our cell we have our organelles, right? So we have the nucleus, we have, say, the Golgi, we have our mitochondria. Ooh, that's a big ass mitochondria, I don't like that. There's our mitochondria, and say we have some other organelles like lysosomes, right? And we can draw all the organelles. The point that we want to get across, though, is that metabotropic receptors are receptors. They're little proteins that exist. We'll do it in pink. We have these little proteins that permeate the lipid bilayer, right? This is not to scale, obviously. I can't make my pen go that thin and that small. However, we have these receptors, and upon the addition of some ligand, right? So we have some ligand, it could be glutamate, right? We call this glutamate. Right, so we'll call this glutamate. We'll talk about glutamate. We'll talk about the exact structure of glutamate. Maybe not. That's more of a chemistry thing. And we'll go into that because I think it's necessary to go into it. But for right now, let's just think of glutamate as this chemical, right? It's a chemical, it's not a protein, right? If a protein is something that is made up of amino acids, and that would be this guy, right? So this is we're going to call it a metabotropic. So we're not going to go ionotropic yet, because we'll talk about ionotropic, but we want to contrast these. So metabotropic receptor. This is a receptor that will take this ligand, this glutamate, and upon the addition of the addition of the glutamate on the receptor, something is it's going to propagate some conformational change throughout the entire protein, right? Now, here's the difference between metabotropic and ionotropic. Metabotropic receptors, they're going to initiate a long cascade of events inside the cell, right? So you could have this propagation of movement or of conformational changing throughout the cell. It's going to reach this end of, or not throughout the cell, throughout the protein. It's going to reach this end of the protein, and when it reaches that end of the protein, it's going to move in some way, right? We don't know how it's going to move, but say it moves into something that looks like, I'll just erase this part and add a color. Perhaps it moves like that upon the binding of the glutamate on the outside. And then, because that moved, things on the inside, right? So things on the inside here, you can have chemicals, you can have proteins. They will say get attracted to this guy, and they will get phosphorylated, and they will attract another protein that will do something special. And you really just get this cascade of events. And in some cases, this is what's called, or not in some cases, but this is what's called or referred to as cell signaling, right? So something from the outside, glutamate, binds to some receptor, right? The receptor, upon the binding of the ligand, is what is called glutamate ligand. We'll write ligand here. Right, upon the binding of the ligand, there's a propagation of movement. So propagation of movement. And so I'll make that in red actually, because that's what our propagation looks like, our propagation arrows. So propagation of movement. That propagation of movement is going to reach all the way inside to this end of the cell, or this end of the protein. And finally, there's going to be that propagation of movement is going to cause the movement of these ends, which is going to activate something, right? It's going to activate a cell signaling sequence of events. Now, typically, and so what can this sequence of events do? Well, the sequence of events can go to the nucleus, right? So, and that could initiate some sort of transcription process. It could say go to another protein that exists on the membrane here, right? So suppose we have another protein here on the membrane, and that will initiate some curvature, say, in the membrane. Right, you can literally think up of anything, right? You could think of say, you get cascading of events that target the mitochondria, or cascading of events that target the Golgi, right? If you are a skeletal muscle, right, you can target, say, ryanodine receptors or something like that, right? So the idea again is ligand binds to receptor changes its conformation, and the conformation is sort of the means by which things start to happen in the cell. Now, what about ionotropic? So I went on this long rant about metabotropic receptors. An ionotropic receptor will, let's see, how do I want to do this? I'll copy all of this. And put it over here. Paste. So what I'm going to do is I'm going to say, erase some of these guys. So here's our cell again, and I'm going to get my green, fix the membrane, get my blue, fix the Golgi. We're back to square one. So an ionotropic receptor, let's make this light green. An ionotropic receptor is going to look like this. Right? So you're going to create a channel that goes from the outside to the inside. So what happens essentially is, okay, say we have our glutamate. Have our glutamate. Glutamate binds. Right? Upon the binding of the glutamate, you will get a conformational change. But the conformational change in this case actually allows for ions on the outside. So like sodium or calcium or say, what's another one? Chloride, depending on the ion, right? Depending on the ion, these guys will rush through here. And depending on the ion, depending on the receptor, right? These ions will rush through the receptors upon when the glutamate binds. That's key, right? Because if there's no glutamate, there's no rushing of the ions into the cell. So you have this rushing into the cell, and then those ions start something in the cell, right? Chloride will hyperpolarize, right? Depending on the nature of the cell, depending on what kind of cell you're looking at, the hyperpolarization patterns might look different, right? Because if you haven't taken this before, we might go into this. The hyperpolarization, again, that's making the cell even more polarized, right? Making the inside more negative, essentially. When you are depolarizing, you're making the cell less, you're making the cell more like the outside surroundings. You could think of it like that when you're depolarizing. When you're hyperpolarizing, you're polarizing the cell from the outside surroundings. And so the idea is that chloride, right, things that have negative charge to them will make cells more negative, thus making them hyperpolarized. If you have calcium, for example, calcium's a very depolarizing agent. Sodium, very depolarizing, right? And we'll see patterns like this throughout, not just when we're looking at glutamate receptors, but when we are also looking at, say, other types of receptors in the heart and so forth. I don't want to go too deep into that, but that's the point, right? So I'll reiterate myself. Glutamate binds. Here, we can go through this again. So glutamate binds. Upon the binding, conformational change will happen, but instead of inducing a cascade of sequence events inside the cell, you allow the rush of ions to flow into the cell. And the rush of ions is what's important because it hyperpolarizes or depolarizes the cell. And upon the hyperpolarization or depolarization, you can get some action potential, for example, or you can get something happening in the cell that you want that the cell needs to have happen, right? When we're looking at skeletal muscle again, we want to contract our muscles, right? And in order to contract the muscle, you need calcium inside the cell to go up so that contraction can happen, right? So calcium enters the cell through channels, L-type calcium channels, for example. And in our case, we're looking at glutamate receptors, and glutamate receptors will allow in some cases, in some type of glutamate receptors, they will allow for the entry of calcium. Some receptors, right, we'll go into this, and this is that paper that I covered in my intro video. For some receptors, calcium will actually block the channel, right? So we have, say this is our glutamate receptor. Just for right now, we'll get into a more detailed picture of what these guys look like. Say this is our glutamate receptor. Sometimes what will happen, we can see, and we'll be able to talk about this even more, is that sometimes the channel will get blocked, right? This guy here is calcium, right? A calcium can actually block the channel. Now, so we've talked about ionotropic, what ionotropic means. And we sort of talked about that in the context of what the effect of these guys are for cells. We can think now, okay, glutamate. Glutamate, all it really is, it's a chemical, right? We'll go over its chemical structure later. I don't really want to go into the chemistry details yet, but it's really just a chemical. And what it does, what it enables us to do, is it enables again the cells to become permeable to ions. And that's exactly what we've talked about. That's all glutamate does in most every case. So we want to get into our heads that glutamate is really just a ligand, right? Now you can ask the question, okay, if glutamate allows for depolarization inside of a cell, what about other chemicals? Can other chemicals act as ligands for these guys? The answer is yes, right? So we'll go over a big list of other chemicals that can affect these channels in different ways, because here's the thing. These channels, they are permeable to, say, there's these class of chemicals called UBP ligands that will bind, and instead of opening the channel, say, I don't want to get too specific, we'll just call it X, ligand X. Instead of opening the channel, I might close the channel, right? So ligands, so the ones that sort of close the channel are the ones that are typically regarded as antagonists, right? So those are the ligands that will render the channels impermeable to ions. Then we have agonists. Those are the channels that those are the ligands that render the channels permeable. Then we have partial agonists. And these partial agonists render the channel sort of semi-permeable, right? So we can actually discover ligands that will desensitize channels faster or slower. So when the channels are in the presence of glutamate, but also in the presence of some partial agonist. If the partial agonist were to bind to the receptor, so the receptor might close even when there's glutamate around, and it might remain closed, right? So these are different types of ligands, and this holds true for all receptors. For all receptors, they have their associated list of agonists, they have their associated list of antagonists and partial agonists. And we'll be able to understand how we can measure the effect of these using electrophysiological methods. We'll do that later. So I'm pushing here on 20 minutes. I don't want these videos to go on for too long. So I'm just going to sort of go through this again really quick on what we've covered. We covered the idea of ionotropic receptors, right? And that was the difference between sparking a signaling or cell signaling, right? That's metabotropic. Those tend to be slower in nature. So I'll write that here. So metabotropic is slower. Tend to be. In some cases, it could be really fast. But on average, or in the gist, metabotropic receptors tend to act slower than ionotropic, right? So this is the ionotropic type, and these tend to be faster, right? Now, what I really want to do in this video is not just go through the biophysics of these channels, which is what we're going to do, but I also want to talk about sort of the distribution of these glutamate channels within the brain, why these glutamate channels are important, right? And we'll take a look at some of the things that you might be interested in, which might be, say, what happens if you get bit by a snake? Or what happens if you get poisoned by a spider or something like that? A lot of times, the reason that you get the effect that you get when you're bitten by a poisonous snake or when you're bitten by some venomous spider or something like that, a lot of the reasons, or one of the big reasons why you get the effect that you get, is because the chemicals and the proteins, the small proteins in the toxins or the venom, will bind to these receptors, and they will affect them greatly, right? And so this sort of those things fall under the list, right? So venom, toxins, and so forth, right? All the way down to not even those things, but like caffeine, for example. Caffeine has its own sort of receptor types that it targets. Anything. This is a huge array. This is essentially what pharmacology is, right? Pharmacology is looking at sort of the function of these receptors in the presence of different types of chemicals and understanding what each chemical, or what the effect is of each ligand or each chemical on these different types of proteins, right? That's pharmacology in a nutshell. But this is, again, so we're starting our discussion on ionotropic glutamate receptors, and with that being said, I'm going to stop at 22 minutes, and I'll see you guys in the next one. Bye.